the types of structures and functions that we see in lipid structures inside the body. So, lipid structures and functions. When I say lipid, what do you think of? Fatty acids, yeah. And when you think of fat in the body, what do you think of? I think of way too much exercise. What? Huh?
Think of the pictures that people say this is how much fat you lost.
Oh yeah, you see the pictures or the gel. Yeah, that I've seen. Oh, Oprah Winfrey one time carried out a little red wagon with all of this gel material that was how much fat she lost. And it was amazing. And really, the loss of fat is a struggle that a lot of people have. It's a burden on the health of America, particularly, and way too much fat that we're carrying around causes a lot of different metabolic changes in our body that challenge how we process and how healthy we are. And so when you think of fat or lipids, what you really are thinking most often is the triacylglycerols, the fat storage. Now, one interesting thing about fat is this. The more fat you have, the less you weigh. Okay? How in the world, that doesn't sound right, does it? To a layman, that absolutely doesn't sound right, because when I get heavier, I have more fat, right? But really, this is how fat allows us to have effectively more energy in our bodies than actual weight. Now, fat is a much slower energy source to access. Glycogen is much faster to be able to extract. But here's the problem with glycogen. It's a storage of lots of sugars. And those sugars have lots of hydroxides. And so for them to be in the body, the carbons with those hydroxides are hydrated and water's attached to them. So for every carbon that is there, there's a number of waters that are with the glycogen molecules. And so when you equate the energy of the carbon, it actually weighs more because it's not just the carbon, but it has all that water associated with it. The same amount of energy, if converted into fat, exclude water because of its hydrophobic nature, and so I get more unit energy in the fat structure than I would per weight than I would per most sugars and even proteins because many of them are charged. And so lipids are very important, but we narrow down and we think about lipids as fat. There are a whole bunch of other lipids that are important to the process. Okay? And so lipids are storage of energy, and that's probably the first thing that we think about. Then, in terms of usage, structural is actually a much larger weight in the body than you probably would even realize. A lot of lipids are cofactors. We've looked at them, and so the portion of the molecule that accepts electrons in FAD is a lipid. And so we'll see some other ones. The electron carriers, all of those are lipids. Hormones, quite often, are lipids. We have several that are intercellular messengers, and then a class called an emulsifying agent, all of which are functions of lipids. So it's much more diverse than we think about. And so lipids can be a whole bunch of different things. Really, lipids are hydrocarbon rich that are insoluble in water. They can have a polar side with a very large nonpolar side, and so they can orient themselves in water. Sometimes they're just really hydrophobic and don't want to be necessarily in the body in the water, and so what we have to have are carriers. So there are a lot of proteins that will carry lipids or structures that carry lipids in our aqueous environment. So you can go back and you can look at lots of different names of fats that we have. And there's a nomenclature where it can show you how many carbons initially, followed by a colon with a number that indicates the number of unsaturations, and then a delta and numerical superscripts that tell you where the unsaturations occur. And we have lots of them. But which fat is most often found in the body? How many carbons? There's one that is way farther in terms of the percentage of that fat found. Not 18. It's close though. It's the 16 version. It's the limit of what we normally synthesize. We can synthesize longer fats, but normally we stop at 16. So most fats are derivatives of 16, either fully saturated or unsaturated. Now, what is unfortunate in our perspective is that even though we see 16 most often, the ones that actually do other things are longer or shorter. And so when we present biochemistry, we show the oddball ones, which might be small percentages, but have a whole lot of functions to do. And so we may get a little bit of skewed view of what's actually in the body. It may be very small in percentage, but it's important in signaling or processing. All right. Should you memorize all of these names? Please don't. You can always look them up. Palmitate and stearate, the deprotonated forms of the 16 and the 18, are ones that you probably need to know. And then as we introduce some other ones, you'll see. Take a look between 12 and 24. Look at the melting points. What happens between 12 carbons and 24 carbons in terms of melting point? 24 has a fairly high, much higher melting point, and the 12 has a much lower melting point. Okay, and so the longer it is, the more interaction that you have. Now, let's go back to GChem 1. What type of intermolecular force, or IMF, is happening between these fatty acids that affect its melting point? Hydrogen bonds. What do you need for a hydrogen bond? Yeah, you need a hydrogen. That's good. And what? You need a nitrogen or oxygen. One, the hydrogen is covalently linked. The other oxygen or nitrogen needs a lone pair. If you go all the way back to GChem, fluorine is in the mix, although in the body we don't have a lot of fluorine. We have one acid group, mostly found in the deprotonated form, and so there is really no proton there because it's deprotonated, and so it's not hydrogen bonding. Oh my goodness. Can you name your intermolecular forces? One is hydrogen bonding. Ion dipole, yes. Dipole, dipole, yes. London dispersion force, okay. In this case, we're really interacting through a London dispersion force. Specifically, GE chemists and biochemists call it different things. A biochemist will call it a hydrophobic interaction, okay. But we're not having a very strong interaction. It is not as strong as, let's say, ion-ion or dipole-dipole interaction. It's just the London dispersion. Now, take a look at the 16 through 20 down in the bottom half. Look at their melting points. What is the difference? They're really low, extremely low, yes. All right, why in the world do those have much lower interactions, well, let's say less interactions between themselves, because it just takes less energy to break them apart? Okay, so they're unsaturated. Well, why does that make a hill of beans difference? What does the unsaturation do? Well, you got to go all the way back to how intermolecular forces can increase. One, they can increase by size. You just have larger molecules. You can have more imbalance, for instantaneous amounts of time, which attract each other. Well, some of these are pretty large, okay, and they have lower melting points than some that are smaller. So it's not a size issue here. The second thing about an intermolecular force, or the London dispersion, is how close you can put the two entities together in terms of how packed you can get them. If it is saturated, it can be a long linear chain in which they can compact really close together. You start putting a double bond in here, and here is the key for naturally existing double bonds. They're in the cis conformation. What happens is they cannot compact or interact as well. And so if they don't touch as often, or they don't interact all the way down the molecule, then you're not making the stabilization. That's why you get melting at a much lower temperature. Here, the saturated versus the unsaturated. All right, so when we make fats in our bodies, we do the unsaturated in the cis. When you buy foods, what are you trying to avoid? What kind of fats? Trans. Why would trans fats be bad? Yeah, they can pack more densely. Normally, unsaturations are going to be good for us. And you can see that in terms of olive oil versus beef fat or solid butter. Olive oil has a lot of unsaturated. Beef fat has a lot of saturated. Okay? If by chance you take in a lot of beef fat and butter versus olive oil, okay, you have lots of fats roaming around in your system, some of which is in the bloodstream at any moment in time. And if you have fats that like to interact with one another, they can stick together and, quite frankly, stick right in your arteries and veins and block them up. And so the more you have unsaturation, the better it is, but you want naturally existing unsaturation in the cis form, not the processed trans. Now, why in the world do we get processed trans fats in our diet? It is because a lot of fats that are used in cooking are wonderfully unsaturated cis bonds. But the problem is, if you have cis unsaturated, it's oily. It's hard to work with. Okay? It would make the food runny. So what they do is they use organic chemistry to reduce them. Unfortunate to that, when we do this industrially, when we reduce them, our processes tend to make the more stable structure, the more stable trans, and so, but it hardens up the material enough to process through the lines, create food, and so we take a very healthy fat that is hard to work with, we make it a trans fat, which is easier to work with and easier to bake with or put into material, but it is less healthy for us. So if we compact them, they can go all the way up to waxy, and if you get those unsaturated in the cis, it becomes oily. We understand lipids as, and we think about this more often, as storage, and so we get a glycerol attached to a lot of fatty acids. Okay, and so where in the world do you think you're going to get glycerol from if you think about metabolism at this moment in time? Where would you grab that carbon chain, that 3-carbon chain? Glyceraldehyde, yes, or dihydroxyacetone phosphate. So that portion of the molecule can come from glucose, and quite often does. Conversely, if I break down fat, that glycerol can actually go and synthesize glucose back. All right. Now, so I have a fatty acid and glycerol react. All right. Let's go to organic chemistry. What is that functional group? An ester. It's great. All right. Esters are stable. Esters are hydrophobic. But esters also can be hydrolyzed by the addition of water, because to synthesize the fatty acid and the glycerol, we have water taken away. And so we'll have enzymes that will create and degrade these ester bonds. Oh, good. Check this out. I asked a question. It's right there. All right. The advantage of fat storage is that gram to gram fat contains twice the amount of energy as carbohydrates. And so we can carry around a lot less weight. Also, in some animals, it can act as an insulation, so it's an additional advantage for them. For us, even though lots of us have insulation, that's not necessarily how we keep ourselves warm. Okay. Now, fats, what I do want you to understand is some non-metabolic uses of what we see and what we use with fats. Has anybody ever gone to a Renaissance fair? And so, in a Renaissance fair, you can buy ye old soap. Okay? Well, ye old soap comes from beef fat, usually, mixed with lye or sodium hydroxide. And what that process is saponification, where we use the hydroxide to break the esters into the fatty acids that have a salt with it and glycerol. And so we can make soap. Soaps are usually very basic because there's an additional unreacted amount of base in the mix. And the reason why soaps are slick is because of the base content, not actually because of the fat. If you actually spill base on your skin, it feels slick like soap. That's where you're getting that slippery feeling from. Now, how do the soap and these fats work in the body? Okay. Have you ever tried to wash your hair without shampoo and you get it wet? Does it get clean? No, not particularly. It gets wet, it dries, it can make it difficult to comb and to brush and to make it look the way you want it to. But it really doesn't take off the material, the dirtiness that you want. What happens is the dirt and the organic molecules, the oils and the greases that you naturally secrete won't be washed off by water. But if we use a single fat that has a polar group, the acid, then what happens is when you mix that into water, they create what's called a micelle, where they put all the fat chains in the middle and they, on the outside, place the acids. So what you get is a sphere of polar and a hydrophobic core. And so as you mix up the soap in, let's say, the hair, what can happen is the hydrophobics attract to the dirt and the grime and the grease, and what they'll do is they'll hydrolyze, or they'll dissolve all the organics inside the organic layer in the middle, surrounded by the polar groups. And then when you rinse that sphere that looks polar, and all the hydrophobics are on the inside being washed away. And so this is how micelles work, and this is how you can get rid of dirt and grease using a soap. Now, if you go back and look at the slides, there are the common names of soaps or fats and the technical names. One of the names for a 12-carboned fat is called laurel. And if you take a look at soaps and shampoos, sodium lauryl sulfate is one of the major components. It's the fat component. And that is used all the time. When we talk about detergents in our labs, we use something called SDS. Have you ever heard of the SDS page gel? The detergent there used is sodium dodecadyl sulfate, which is lauryl sulfate. It is the main component of shampoo. So our SDS is basically what's in all shampoos. Now the metabolic uses that we're going to use fats in this are we're going to take fats, and from dietary fats we're going to send fats around in kilomicrons. Okay? From the liver we're going to send around fats as VLDL particles. And if we have the fat cells mobilizing fat to be used for exercise, we're going to move around the fat chains in the form of albumin. All right. Anybody taken biochem or cell lab yet? Okay. At one point, you have to have a protein standard. And so you compare an unknown amount of protein to a protein standard. One protein standard that we use quite a bit is called BSA. Does anybody know what that stands for? Bovine serum albumin. The blood system has a large concentration of protein called the albumins. And it's in the serum, and it's really easy to isolate because it's already solubilized in a liquid, and so it's easy to take that particular protein. That protein is used as a standard quite often. Well, we also have an albumin. We call it HSA, human serum albumin. That protein roams around in the bloodstream. It has very large hydrophobic pockets such that a free fat can bind into it and be carried in the bloodstream. Because a free fat doesn't really want to be dissolved in the water. It can carry up to 10 fats for every albumin. And so we've got to move these things around in ways in which these very hydrophobic entities are being able to be soluble in water. So when we eat dietary fats, okay, so this next slide right here is basically after you've gone to Taco Bell and had fourth meal, okay, you have an influx of a lot of fat, okay? And what's happening is by the time we get into the intestine here, the fat is still fully formed. It is emulsified or dissolved by bile salts, and they're carried around to the intestine. At the intestine, we have an enzyme called a lipase, which actually breaks those ester bonds into the free fats and glycerol. The free fat and glycerol are entities that can be transported. The triacylglycerol cannot. So we break it down so that we can bring it inside the cells. Now, unfortunately, we've spent this time to break it down. We will resynthesize the fats into triacylglycerols and then package them into a carrier molecule. And so it's a small vesicle that has a plasma membrane, it has proteins on it, and inside we put the fats. And so we'll excrete that particle and send that into the circulatory system. Most of this will be absorbed by the liver to be reprocessed. Some tissues can take this directly and take the fats in. Those particles that, taking a little bit better look at that, you see that inside we have the triacylglycerols, the fats, but there are other fats in there, including cholesterol that are esterified, the hydroxide. We've got proteins that will be proteins on the cell surface that will dock with the cells it needs to dock with and cause function. The membrane is also made up of lipids, and these particular lipids are phospholipids. We'll take a look at those structures in just a moment. And there's also free cholesterol in those particles. And so they're mobilizing, moving around. They will bind to cells using those particular proteins on the cell surface, activate lipases again, and then the free fats get brought into the cell, The glycerol stays in the bloodstream and then taken to the liver. And then those fats can either be used by that particular cell or moved through the body in carrier proteins, albumin complexes. The cells will be taken up, and inside the liver, you've got all sorts of different things happening. You can take carbohydrates, make glucose, make pyruvate, make acetyl-CoA, and you can make fats. We'll do that later. You can take proteins, and a lot of the carbon chains of proteins can be made into fat. You can take glycerol and make fat, and in the liver, we can compartmentalize all of these things, put them in a package called a VLDL particle, and we send it out into the bloodstream. VLDL, have you ever heard of that particle before? Yeah. Now, a lot of times we use terminology in terms of LDL and HDL with what kind of disease? What is it? Cardiovascular. What we're looking at in terms of LDL and HDL, which is high-density lipoproteins, and LDL, low-density lipoproteins. What's VLDL? Very low density, okay? The other two, we're talking about how much cholesterol is in the mix. There's cholesterol in this particle too, but the cholesterol concentration versus everything else is not so bad. The LDLs have such high concentrations of cholesterol that is worrisome to our circulatory system. The HDL has very low, and so when we look at cholesterol concentrations, we're not actually looking at concentration of the molecule cholesterol, we're looking at the concentration of the carriers of cholesterol. And we're looking at LDL versus HDL, and we look at a concentration per deciliter, and we have to be below a certain number between the two, and I believe that's about 200 milligrams per deciliter. And then we're looking at the distribution. You could be under 200, but you might have the wrong distribution. You may have way too many LDLs versus HDL, and so you can have disease in two different ways. Okay, in terms of mobilization, what really happens is we get all of these triacylglycerols and we compact them into storage cells called adipose tissue. And adipose tissue just really collects them all into these gel-like drops that sit inside the adipose tissue. There's not a lot going on in the adipose tissue. It's just a lot of fat that just sits there. Okay. Unfortunate for you is this: if your body makes a brand new fat cell, that fat cell will be with you forever. You don't get rid of fat cells. You can decrease how much fat is inside that cell, but you have that fat cell for life. Okay. We only send fat cells signals to mobilize when you get a glucagon state or an epinephrine state. In this, we will get glucagon, we will start phosphorylating lots and lots of things using pKa, and that will mobilize hormone-sensitive lipases to start breaking these down into fats, and the fats are then transported into the albumin, and those albumins are taken up mainly by muscle tissue. And so, again, if you're trying to lose fat, you need to exercise long enough to go through the glycogen stores, which you can exercise for quite a while on your very quick sources of energy, so that you get to a metabolic trigger to start sending these particular molecules out. And so very short workouts may help you in this. You won't store any more fat, but you may not burn fat. You have to get longer exercises to do that. So glucagon and epinephrine will affect the adipose tissue itself. Now, we'll talk about how we break those down next chapter, once we have those free fats and how we actually degrade those, and then what the energy is converted into. We've introduced, and you've seen lipid bilayers all over the place. We're now going to take a look at what is the actual components of the bilayers. They're broken up into a couple classes. One is phospholipids, the other is sphingolipids, and the other is cholesterol. Okay? Both the phospholipids and the sphingolipids, okay, have a general structure of something polar and two hydrophobic tails. And so a lot of times they're kind of drawn like this, something polar and two tails. And you'll see a cartoon like that all the time. Now, in terms of that, we have to add the equivalent of two fats to the binding molecule. And to make it polar, we're going to add on really hydroxide-containing groups most of the time. And so one has a phosphate in it, phospholipid. The other doesn't have a phospholipid on it, but it has a base molecule called sphingosine, and we get the sphingolipids. Cholesterol is an oddball in this case. It has a very hydrophobic portion that can fit into membranes really, really well. Its polar group includes a single hydroxide. So its polar head group, yes, is polar, but it's just one tiny little hydroxide. It's enough to orient it into the membrane. When you take a look at the membrane, there's all sorts of things that are incorporated, including, you see that little ball and two squiggly lines? All of those phospholipids and sphingolipids. There are also proteins that are embedded. There are also sugars that are embedded on the outside. And so it's fairly complex. And so if you take a look at any particular cell class, you're going to have a different percentage of all sorts of things embedded in that membrane that cause function. And so the largest usual section of lipids is the actually phospholipids that are there, and then it breaks down into cholesterol and sphingolipid types. And then the proteins, depending upon what you're doing, can be a whole bunch of proteins in them, like let's say the membranes of the mitochondria, or you can have some that have absolutely almost no proteins, depending upon what you need to have done. Now, proteins can be embedded or associated to the membrane, and so let's take a look at the animation on how we can see these proteins in the membrane itself. So a plasma membrane essentially is a barrier. What we're trying to do is make two different things across a membrane space. We spent a lot of time creating a proton gradient. From that, that's a form of energy. We only do that because the membrane prevents protons from moving across, unless we have a protein that sends them across specifically for some sort of purpose. And so we've talked about lots of proteins. They can be pumps, channels, receptors, energy transducers, enzymes, all sorts of things. The proteins can be embedded, called integral, or associated to the membrane, called peripheral. The proteins also are oriented, and this is very important. If you take a look at the ATP synthase in both photosynthesis and oxidative phosphorylation, they must be oriented such that the channel for the protons are where the concentration forms, so that it flows through that particular direction to make ATP. If you had the ATP synthase both directions in the membrane, then as soon as you break the gradient, then you'd have one flowing the opposite direction, and you'd be making ATP in the wrong compartments, and it really wouldn't work. So not only are the proteins themselves identified for a particular process, they must be placed inside the membrane in a certain order. And I told you that most sugars are presented on the outside. We have to make sure that the proteins that hold onto the sugars are on the outside of cells, and so the orientation is very important. The types of fats that are incorporated into membranes, okay, when we take a look at them, are all over the place in terms of what is there, the specific types. Okay, and so there's a whole lot of names on here: phosphatidylinositol, phosphatidylethylamine, phosphatidylserine. At this moment in time, I haven't shown you what those particular structures are. We will learn those. But they're all over the place in terms of what in the world, which cell type it is. Unfortunately, we do not necessarily truly understand why we have to have differences. There are some lipids that are part of processes that must be there for signaling. But the diversity that we see is not fully understood. We have identified that they are diverse, and we're still trying to figure out why in the world or what advantage the diversity provides for each particular membrane. The membranes themselves are fluid, okay? Meaning that there's a lot of flexibility when we talk about the membrane. They are two-dimensional, and they are solutions of proteins and lipids. So we are oriented and we are solubilized in the organic segment. Now, solubilized means that things can move through the membrane. The membranes can actually deform. The proteins that are embedded in them, for most cases, can actually diffuse through that. And they usually can move laterally around, but they do not flip. They don't go from one side to the other. If you take a look at what's happening, you see that big old protein in the front. It's a big complex. It's moving around. There's energy involved in the cell, and it can move. But there's a little protein in the back that's whipping around. Some proteins are very mobile. Some proteins move a little bit. And it's usually based upon size. But there are some proteins that you see with those sugars that are linked behind the scenes so that they are embedded in a place that is specific. And so some proteins can move, some proteins don't. How I make the protein more fluid or less fluid is essentially adding more unsaturated versus or more saturated lipids inside that membrane. And they're always moving around. You can see the effect if I actually take a membrane and you heat it. The dance floor gets busy, okay? And you can actually break a cell by just heating it. It's cooking. You're breaking the cell membrane down, breaking the integrity, give it enough energy. Now, putting cholesterol in to a membrane, all those fused rings, prevent saturated fats from getting together, so it lowers the fluidity. But if you had a lot of unsaturated fats, then putting cholesterol with those fused rings makes it more rigid and increases, makes the membrane less fluid. And so by adding cholesterol, I'm going to make it more fluid and less fluid? Yes. The answer is absolutely yes. Cholesterol does both. Cholesterol is a modifying or a moderating. It makes it a little bit more fluid, but not too, because if it's too fluid, it'll make it a little bit more rigid. So it keeps it in the middle. We know that, and we've taken a look at a lot of proteins that are embedded. We talked about pumps, we talked about channels, we talked about active and passive. And so we've talked about the proteins and their functions, including oxidative phosphorylation, where it's all happening in proteins in the membranes. So we know that there's function there. In terms of ones that have almost no protein is myelin. Myelin is a nerve cell, and what we want to do is have ions move across, but very controlled. We only have a few proteins that are the channels that allow those ions to flow through. Most of the time it's very hydrophobic, so nothing else gets through. Membranes, in terms of energy transduction, or like the complex 1, 2s, and 3s, the protein content of the membrane is almost 75% protein. Most cells have about 50% lipids and proteins. Integral, embedded, so A, B, and C are the integrals. Peripheral are associated, D and E. The integrals can be released from the membrane a lot of times by just changing the pH or salt concentration, because they're being held together by interactions with and to the integral proteins. There's another way that peripherals can actually be associated. All right, here's an experiment that I wanted to show you, and I want you to get an understanding of what we do in lab and what we see in a book, okay? A book usually gives you this wonderful picture about what is happening in the experiment, and then it gives you at the bottom here what the data might look like, okay? And so this particular experiment is going to take a group of the same cells, and I'm going to react it with two different reagents. One reagent that can cross the membrane and react with proteins, and one reagent that cannot pass the membrane, but it does react with proteins. Okay? If you were doing this experiment, you wouldn't have this big picture. What you would see is this. Okay? And you would know that this reagent can go through and that reagent cannot. So in the first lane, with a reagent that cannot go through the membrane, the analysis of interacting that reactant with the cells, I have one protein that is on the cell surface that I can modify, and another protein on the cell surface. One relatively small, one relatively big, if I look at separations by SDS page gel. I compare that to a second reagent with a second sample, and I see a band here that represents P3. Now, if I have a reagent that can go through the membrane, it's going to react on the outside and the inside. Okay? And then I pick up a protein here, and I pick up another protein at the top. All right. When I compare this, I can say, okay, this protein is accessible on the outside. This protein is not on the outside. This protein is on the outside. What can I say about this? Obviously, this protein is only on the inside. It doesn't actually get all the way through to the outside because I don't see any reaction here. Now, how can you interpret this bottom one, this P3, versus this P2? Okay, what is different between this band at the bottom and this band at the top? The top is thicker. Okay? Now, here's the deal. Between this sample and that sample, this looks darker than that, right? So you might say that this was more labeled. Okay? But if you take a look at this one and that one is similar, right? But these two have a certain amount of reactivity. That one's a whole lot more, right? It seems to be a 1-to-1 to much more ratio. Now, if you didn't have the picture, and what you now can see in the picture is that this protein gets modified on the inside because it has a surface that touches on the outside because it has a surface that touches the outside. It will also be reacted on the outside, but it's darker. So that means it must have a surface that can be reactive on the inside also. It gets double labeled. Okay, there is the 3. And in the case that I'm only on the outside, I only get it once. Here, in protein 2, This gets labeled with the stuff on the outside. This gets labeled with the stuff on the outside, but it's like in a 1-to-1 ratio, and so P2 must only be exposed on the outside. It doesn't actually go all the way through. And so, sorry, is that P3? P3. And you can see in the picture, P3 doesn't actually go into the inside. It's only accessible to the outside. And then P, is that one? 1 is only on the inside, and you can see that the protein is only accessible on the inside, is not on the outside. Books tend to do this. Lovely picture, and maybe tell you what the experimental data would be. Okay, sometimes they don't even do this. But in real life, you do an experiment when you're doing research, and what you have to do is you have to take this data set, and then you have to describe in your paper or put a figure in your paper that explains the data that you see. And so teaching this arrow is much more easy than what you're really going to be expected to do is to take something like this and tell others that's what's happening inside the cell. Now, this slide has nothing to do with what I'm going to ask on the test, but I do want you to understand that as you learn everything, it's the known. And what we want you to be able to do is to interpret or to actually estimate what the data is, but eventually, as you do more research, as you do more experimenting, as you know more, what we ultimately want you to do is be able to go backwards. Okay. Now, how do I get proteins out of the membrane? Well, usually we take the membrane, and it's really hard. It's more of an art than a science. But what we can do is mix up the cells with some detergent. What we want to do is replace every organic interaction or hydrophobic interaction that was in the membrane with a detergent so that now it can be completely solubilized. So proteins that are embedded have wonderfully hydrophobic interactions here. Polar there, that wants to be in water. That part wants to be in water. This part wants to be in water, but this part doesn't want to be in water. If I mix in some detergent, I allow the hydrophobic tail to coat where it is hydrophobic and put a very polar molecule so that the whole protein is solubilized. Unfortunately, remember way back when we had SDS page gels, we put in SDS to denature a protein. So too much detergent will destroy the protein. Not enough detergent will not get it out. So you have to find the Goldilocks amount to actually pull that out. How do you find that? You do research, meaning that, what does research really mean? Well, when you go into GChem, you don't do research, you do a search. We give you the procedure, you're supposed to do it, and it's going to work out, unless you mess up. But research is this: you may not have a procedure, so you got to do it. If it doesn't extract information, then you got to redo it and redo it until you find a combination. And so it's lots of experiments, and research is expecting that you're going to have to redo until you can confidently say that what you did produced this data, and it will always produce this data, and that data means something. Peripheral proteins are attached normally and a lot of times through non-covalent interactions like ion interactions between an integral and a peripheral protein. I can just use some salt and knock those off. Other peripheral proteins can use fats to attach them. And so this huge thing right here is attached by two tiny fats in the membrane. It is not that much stability. It's enough to actually embed it, but a little bit of strain will knock that thing off, but it's considered a peripheral. Integrals are really embedded in the membrane, and only books use these kinds of types things. I don't want you to know the types, but this is what I want you to know, is that a lot of times membranes that have proteins have a reoccurring theme. 20 amino acids that are in an alpha helix that are hydrophobic. If they are 20 amino acids long, they tend to form an alpha helix and are hydrophobic. That is the exact span of the hydrophobic nature of most membranes, because it's 16 carbons to 16 carbons. When you get 20 amino acids curling, that's the same distance. So if I have a primary sequence, I can look for places where hydrophobic alpha helix are, and they could potentially be membrane incorporated. Unfortunately, this also can happen. It is as highly likely that that alpha helix will just be on the center of a protein, because as we fold, we put all the hydrophobics on them in the middle. Although most amino acids that are in the middle are 14, 16, 22 different numbers. If it's 20, that's where you get the idea that it might be. Quite often we can plot the value of its hydrophobicness against its sequence, and we can get a hydrophobicity plot, and you can actually identify the potential candidates in which membranes can be bound. You can embed proteins by a single alpha helix, but there are some that are combinations. 12 is one that is seen quite often. 7 is another one that is seen quite often. How many alpha helix bundle together to bind into a protein? What helps us with this analysis is knowing lots of other proteins that are embedded that we know. If they have the same pattern of hydrophobicity, it may be the same structure, it may function the same way. And so the more information we get about real structures, we can use analysis through the computer to identify. For a long time, we found lots of proteins that were embedded in the membranes through alpha helix because it was easy to identify. Unfortunately, that kind of put blinders on us about how proteins fit inside the membrane, and for a long time, we missed an entire class, ones that have beta sheets. And so the beta barrel is a moiety that now we see is a common structure. Lots of examples can be seen. In particular, I want you to take a look at this one. Here's the top view of that. You can see that beta barrel makes a wonderful hole. And if we're trying to transport something through a membrane, a hole is a great thing to get through. And so there is quite a lot. And even this, this one really kind of could have confused us. There's a lot of alpha helix in the portion that's soluble in the solution, where the beta barrel is the one that's embedded. So we have lots of ways to get through the membrane, although the beta sheet structures are relatively new in our identification. Now, we have lots of proteins that are embedded into the membranes, and they can move around. And for a long time, we thought that most proteins were just kind of distributed around. And so each cell type had certain proteins that caused that cell membrane to work in a certain way. But after really looking at it, part of the regulation is not just having the proteins there, but having them in close proximity. What we find is when we look at scans of the membrane, we can see that there are places where it's a little bit thicker. And we tend to find these and call them microdomains or rafts. What we see is protein complexes are concentrated in those areas that bring everything together, that help in regulation just by proximity. And so these organizational tools really create regulation. And so just by them being put together, we now also have discovered inside the cells, we also thought all the proteins were just floating around, but we have anchoring proteins that bring whole pathways together so that they're bound together. And again, that's regulatory. I don't know why I put this slide in here. It's basically saying the last thing. The rafts aggregate functional groups. Now, although those membranes are membrane proteins, membrane lipids, really are a very large fraction of where lipids reside within the body. If we take a look at the phospholipids, we have a backbone of either glycerol, 3 carbons with 3 hydroxides, in which we add 3 different things, or sphingosine, which has places in which we can place fats on, but also an incorporated fat that is one of the fats that is part of the structure. If you take a look at phospholipids, we can either attach a phosphate to a glycerol or a phosphate to a sphingosine, and we can add some lipids to it. And so in this case, phosphatate, or we'll call it phosphatidyl, has these components. It has the glycerol as the base. Okay, one hydroxide is bound to a phosphate. The other hydroxides are bound to esters. The type of fat on there can be variable. Most often, it's 16 carbons long. Okay. Once you have this, we can then put a derivative on the phosphate. The phosphate's polar, but what we do is we normally add an alcohol to create a polar group. And so we can add serine, ethanolamine, choline, and inositol, the sugar. Kind of oddball. The projector doesn't show it real well. That's a positive charge. It looks like it's just a line. But, and then the choline also has a positive charge. So they are very polar in terms of even having charge. The fats are from 14 to 24, although most often 16, and they can be saturated or unsaturated. These are the polar head groups. You should know these structures enough to recognize them, but not necessarily to draw them. But these are ones that are attached most often to phospholipids. And so you've got the nomenclature. Phosphatidylserine is 2 fats, a glycerol, a phosphate, and then the alcohol serine. Phosphatidylcholine, phosphatidylethanolamine, and phosphatidylinositol. That's the nomenclature. And you can see that it's just fat attached. We don't even identify which ones they are. Okay? If you take off the polar group, it's phosphatidyl or phosphatate. If you take off the phosphate, you get a molecule called DAG, which is diacyl, 2 fats, glycerol. Okay, so diacylglycerol, you may have seen that before, DAG. And so we can have DAG, phosphatidate, or phosphatidyl, and then the alcohol names. There are a couple of phospholipids that have a difference. They're called they're ether classed. And so they have a ether in place of an ester. And the synthesis is a whole lot more complex. And we're not sure why that particular bonding is made, but there are 2 molecules in this class that are very important. Plasminogen makes up about 50% of the phospholipid in the cells of the heart. Okay, we find it there, and it's a ether lipid. And then the platelet activating factor is a signaling molecule in blood clotting. If you lose that particular molecule, you become one version of a hemophiliac. So these are just alternative ways to add a fat group to the glycerol. And we show these 2 We don't know why the ether is more advantageous than the ester in these particular molecules. In terms of the phospholipids that are sphingosine based, all right, you can see again a polar head group and two fat chains. One of the fat chains is part of the molecule that is going to be added, the derivative is added to. We will add one fat to it to get the two tails, and then we will add the phosphate and then whatever polar head group we want. Okay, and so we have different classifications of these. If you have a phosphate, the two alcohols you see most often, almost exclusively, are choline and ethylamine. Okay, those are sphingomyelin containing groups. If you add one sugar on, the class is cereboside. And if you add on, most of the time, a ganglioside is a lot of sugars, and most of the time there's an acidic group on there to make a ganglioside. Now, when you think of cerebroside and ganglioside, what tissue type do you think of? Brain. They're usually associated with nerve cells. So in some way, the sphingosines are associated with membranes around nerve cells. Glycolipids have lots of sugars, and again, cereboside is one sugar, a globuloside is multiple sugars, and then a ganglioside usually has multiple sugars and charge. And so you can have different classes of them. And then for the derivatives of the gangliosides, you start getting names, and so GM2, GM3, GM4, they get complex, but they don't give you a lot more information than just an acronym. We've already seen an example of this in the OAB type. Those fit within that particular category. But we won't test over this since we've already tested on that. Just wanted to let you know that it fits here. Now, membranes are synthesized. Also, membranes can be broken down. The enzymes that break down these particular membranes are the phospholipases. A lipase was one that used to break down fat, all triacylglycerides. Okay, phospholipases are the ones that contain phosphate. There are 4 versions, phospholipase A1 and A2, that breaks off the first or second fat. Phospholipase C, which would break off the polar head group and the phosphate, or phospholipase D, which breaks off just the polar head group. Phospholipases are found in the cytosol and in some venoms. What actually happens if you get bit by a poisonous snake, there are 2 real issues with that. One is the venom itself, usually a neurotoxin. Not good for you. But the other thing that can happen in either a snake bite or like a spider bite, where they actually inject you, they also inject these phospholipases. And so what consequence that can have is that the cells are degraded and you get just decomposition. And so you might not be hurt by the venom, but the phospholipase may eat away at the membranes themselves. They're a potent combination because they want the venom to get inside your bloodstream, and as they break down the cell walls, the venom has a better chance of getting inside, rather than just being a hypodermic needle that has to interact. And so, yes?
Do phospholipases also come out in like dry bites, what they call?
Mm-hmm.
Okay.
Yes.
You'd still be affected even
Yes.
if you didn't get venom.
Correct. And even if you didn't get venom, this is probably one of the worst aspects of a bite that most people don't even know is an issue. You may have seen in signal transduction phospholipase C as an indicator. It is part of a informational pathway for signal transduction, and it breaks off a membrane-bound lipid, and it sends off, if you remember, it takes inositol triphosphate and makes IP3 and diacylglycerol that will The IP3 goes to the endoplasmic reticulum, lets out calcium. The DAG goes to the protein kinase C, and the calcium and the DAG attach to protein kinase C, and that will phosphorylate and cause activation. So you might have seen that.
Shouldn't just have more phosphorylate?
Yeah, they have all group of them together working, and so it just kind of melts the membranes down. We do have phospholipases naturally in the cell for turnover, so we have all of those there so that we can recycle, but phospholipase C is particularly upregulated in a signal transduction pathway.
So if a patient comes in with a dry bite, how are you going to mitigate?
Oh yeah, how do you mitigate?
'Cause, like,
You know, that's a great question. I have never looked that up. With venom, you give them the antitoxin, right? There has to be inhibitors. I bet you you could do something as simple as washing it out with acid or acidic solutions or basic solutions, because enzymes are always dependent upon the pH. So most likely that would probably be the first treatment. I don't think we would give them inhibitors particularly. I think we may just try to change the pH value so that we detoxify it that way. Cholesterols, we think about it as being a bad thing because we eat too much of it, but cholesterols are really important in lots of different components, including just embedding it in the membrane to make sure that the fluidity is correct. But we're going to, from that steroid, we're going to derive a lot of hormones from it and have signaling happen.